Welcome to MiceTech Talk with the technical information scientists of the Jackson Laboratory. My name is Dr. Caitlin Gilland, and I'm here with the entire MiceTech Talk team today. Our job as technical information scientists is to serve the research community by answering all of your mouse-related research questions. Did you know that the Jackson Laboratory has the largest selection of mice for COVID-19 related research? We were able to obtain our first mouse model for COVID-19 in February of 2020 in the form of sperm, and by midsummer, we were able to produce cohorts of hundreds in size and were able to distribute them to hundreds of labs worldwide. Since then, we have expanded our portfolio of mouse models for studying COVID-19 to 12 different strains, and our portfolio is continually growing. Today, we're going to chat about some of those different COVID-19 mouse models in our collection. So for those of you who are watching live, you can use the comments section of the video to tell us where you're viewing from, if you're conducting any COVID-19 research, and any related research questions that you have, and we'll try to get those answered for you today. So hopefully you're ready with your questions because it's time for MiceTech Talk. So today, let's talk COVID-19 mouse models. Dolores, why don't we start with you, because I know you wanted to talk about the original COVID-19 mouse strain that we have here at the lab.
Absolutely. So the first mouse model for COVID-19 we made available for the scientific community was the strain known as K18HACE2, stock number 034860, donated by Dr. Stanley Perlman from University of Iowa. These transgenic mice express human ACE2, which is the receptor used by severe acute respiratory syndrome coronaviruses to gain cellular entry. The human keratin 18 promoter is what directs the expression in epithelia, including airway epithelia, where infections typically begin. In fact, multiple independent investigators testing these mice have found that intranasal infection with a concentration of 1 million PFUs of SARS-CoV-2 virus results in K18HACE2 mice with severe illness 5 to 8 days post-challenge. Infected mice develop progressive weight loss and lung disease with robust virus replication around 2 days post-infection. The lung disease includes edema, inflammation with mononuclear lymphocytes, macrophages, and neutrophils, epithelial cell death, and interstitial disease. These mice also develop CNS involvement with peak virus replication at 6 days post-infection, which is characterized by neuronal infection. Virus titers are detected in several organs, including the lungs, brain, kidney, liver, and spleen. And on top of that, mice infected with SARS-CoV-2 also replicate cytokine storm observed in many patients with COVID-19. So this data altogether showed that the K18HACE2 mice provide a useful model for studying the pathological underpinnings of both mild and lethal COVID-19 and for assessing therapeutic interventions. The strain is on Black 6J genetic background, and for those of you who prefer to work with models on the bulb C genetic background, we have good news. We also have strain stock number 035247, which will be available very soon, and this has the same exact transgene, but on bulb C background.
So this sets us up nicely to talk about the next model that I wanted to discuss using the same K18HACE2 mutation on an NSG background. But before we move on, I just wanted to give you a reminder to go ahead and submit your questions. I see we have Brian Long viewing from Insight, so hello and welcome. So Xingtong, why don't you tell us about the NSG variation that we have?
Yeah, sure. So I will introduce you today about the NSG K18HACE2 mice. So you can tell from the nomenclature of this strain, it contains two components, the K18HACE2 transgene and the NSG background. So I would like to highlight the importance of this strain from the following aspects. So first of all, this strain has the same transgene mutant allele of its B6 Transgenic K18H2. So it well also expresses the receptor, the H2 for COVID under the control of the human keratin 18 promoter. And in addition to that, because of the presence of the SCID knockout mutation, the interleukin-2 receptor gamma chain knockout and the non-genetic background, this strain has a greatly reduced immune responses and increased susceptibility to the virus infections. So most importantly, it allows you to engraft the mice with human, either the hematopoietic stem cells or the peripheral blood mononuclear cell fractions. So you could investigate the potential therapies or prevention strategies in a more clinical relevant humanized platforms. And the second point would be this strain also offers you an opportunity to generate a human immune system with majority T cells, B cells, and still be able to be infected with the COVID virus. So it can serve as a clinical relevant platform to test maybe the COVID-induced cytokine storm, also called cytokine release syndrome. But again, you might need to do a pilot study. So first to testing the feasibility, the optimal virus dosages, and also even the optimal experimental conditions to create the cytokine storm. By the way, I also want to mention that we can also offer the PBMC humanized platforms. And when you treat these mice, the PBMC humanized mice with your desired immunotherapies, it might also allow you to test in the cytokine release syndrome to strategies to even mitigate the cytokine release syndrome with your desired strategy. So last thing, actually, I would think is more like a reminder that for this particular strain, we generated it by breeding the B6 K18H2 with the NSG mice. We used a SNP panel assisted breeding strategies to crossing these two strains together. And we normally need to breeding for more than 5 generations. And right now, I think we should have this strain ready very soon after completing the last round of breeding. Again, I want to remind you that the genetic background change itself could cause in the phenotype change if you compare the phenotype of the NSG K18H2 with the B6 version. So I would still recommend for pilot studies when you're trying to conducting your study with a large number of animals. So that's all about the strain NSG K18H2 mouse model. Speaking about humanization, Janine, would you tell us more about your models?
Sure. Thank you, Shengtang. So I'm going to highlight a variation of these K18ACE2 strains that we've been talking about. And the humanization I'm talking about is a humanization of ability to look at PK. And that can be used to develop monoclonal antibody therapies and test for efficacy and clinically relevant half-life. So you guys might know in the last month or so, the US FDA actually issued an emergency use authorization for 2 monoclonal antibodies to treat COVID. One of them is bamlanivimab and the other one is atezolizumab. And these 2 antibodies bind to the spike protein and are designed to prevent viral entry into the cells. In clinical trials, this combination therapy was actually able to reduce hospitalization and death by about 70%. But those press releases don't actually go into how you determine the appropriate dosing in patients. And a lot of that work and some of it's done in mouse models as well as human models. But that determination of half-life is one that I wanted to focus on. So generally, things like monoclonal antibodies and IgGs have a pretty long half-life in the serum of about 20 days. And it's beneficial to the patients because it means they don't have to be dosed as often and the treatment is more likely to be efficacious. The reason why the half-life is so long for IgGs is because of a receptor called the neonatal Fc receptor, or FcRn. And this is a receptor that's primarily found in endothelium and leukocytes and grabs onto the Fc portion of the IgGs, rescuing it from lysosomal degradation and then returning the IgG back into the circulation. So the tricky thing about monoclonal half-life determination occurs when you're using a human IgG and then testing that in a mouse situation, or in a mouse model. The human IgG doesn't bind to mouse FcRn with the same affinity as human IgG binds to human FcRn, and that has really negative impacts on your clinical relevance when you're trying to determine half-life. So back in 2001, Derry Rupenian's lab at JAX developed a mouse strain that was a knockout of the mouse FcRn and then express the human full-length FcRn. And since then, there have been a lot of studies that demonstrate that the half-life of these humanized FcRn more accurately match the known human half-life data. Notably, the correlation is better than in standard mice, and it's pretty comparable to non-human primate data. And so that's pretty fantastic from an efficacy and welfare standpoint. So the result is now a platform that pretty accurately predicts human half-life of human IgGs in vivo and potentially can reduce the amount of testing that has to be done in non-human primates. So basically what's been done for this particular strain for COVID applications was to combine the FcRn humanization and the humanization of the K18 ACE2 transgene humanization. And so now in theory, you can infect the mouse with SARS-CoV-2, it'll get sick, and then you can actually test monoclonal antibody therapies with clinically relevant half-life in the presence of that monoclonal antibody target, which is huge.
Awesome. Thanks, Janine. I saw that we just had another question come in asking about stock numbers. I'm not sure if you're referring to the NSG or the FcRn, so I'll give you stock numbers for both of those strains. So the NSG ACE2 strain is stock number 34901. And then the FcRn model is stock number 034902. All right, so now that we've talked about some of the more popular HACE2 models, as well as what it might mean to humanize the mice, let's move on and talk about some more specific mouse models. So Paige, why don't you tell us about the mouse model that we have for studying SARS-CoV-2 infection within the brain?
Yeah, thanks. So, while COVID-19 is generally kind of thought to be a respiratory disease, further research has really started to show that there's a number of neurological complications associated with this. And so you may know that these kind of can range from headaches, loss of smell, tingling or numbness of the hands and feet, confusion and memory disruption, and even seizures or stroke. So there is some work being done through the NIH to start to track these specific neurological symptoms through the NeuroCovid data bank. And so that's the clinical portion of it. But not a lot has really been done yet to address this kind of neurological portion of the disease in the animal models. There has been some work done with the K18 HACE2 model that Dolores already mentioned, and that examined the neuroinvasive potential of SARS-CoV-2 in vivo. And so basically one of the limitations of this K18 model is that it exhibits both the neurological and the respiratory aspects. So this can make it really difficult to start to tease apart the consequences of the SARS-CoV-2 infection on these very different organ systems. And so one way that we can really start to get to the bottom of these neurological symptoms and to tease apart the respiratory from the neurological portions is to use a mouse model that has CNS tissue-specific expression of HACE2. And so there already is a model readily available, which is the SYN HACE2. And this model is a little bit more unique because the expression is driven by the specific rat synapsin or SYN1 promoter. Mice from this line have been found to have high levels of expression widespread throughout the brain and central nervous system, and no expression in the other peripheral tissues that have been tested. So these mice were originally developed in 2010 to actually study the overexpression of angiotensin, and it was found that that's protective against the development of neurogenic hypertension and heart failure, which was induced by Ang2 infusion. And so while these SYN ACE2 mice haven't yet been used for SARS research, I'm really excited to kind of see where this neurospecific work will go and how this model can be leveraged to understand the play between COVID-19 and the brain.
All right, thanks, Paige. We just had a question come in asking if we see any brain injury in these mice. Do you know if there's any data surrounding that at this point?
So that's a really great question. Again, the SYN HACE2 model hasn't been used yet, but the work in the K18 HACE2 model has started to show that there's actually some vascular implications even in the brain. And so that can even be some remodeling of the vasculature. So it will be interesting, again, to see what more research and more neurologic specific research shows.
Great, thanks. The stock number for that strain, let's see, do I have it handy, Paige?
I didn't grab it, I'm sorry.
You have it?
No.
Oh, I see it. It's 399. All right, Sarah, why don't you tell us about the Tempras2 mouse model next?
Absolutely, yeah. So we know ACE2 is a really important critical receptor for a COVID-19 infection, but there's also a lot of data showing that there are other proteins, other receptors that are really critical for some of this infection. And one of those is the Tempras2 gene. This is a protease that co-localizes with ACE2 in the cell membrane, and it acts to cleave the spike protein and effectively primes it for entry into the cell. So Tempras2 has been suggested as a possible target for treatment through the use of different inhibitors. And this is some work that's been done actually originally for SARS-CoV-1 back in 2004. There was some data coming out looking at the interaction between Tempras2 and ACE2. And now with SARS-CoV-2, there's some more work that's being done that's looking at those interactions. Some of the data has suggested from in vitro studies that there is this dependence of SARS-CoV-2 on Tempras2 activity, and that with suppression of, with treatment with a Tempras2 inhibitor, you do see some suppression of infection. However, I should say that when I was looking up some of this data, I saw a few conflicting reports, and it does appear that there are other proteases that can act in the absence of Tempras2. So ACE2 still seems to be the more critical protein, but Tempras2 definitely acts in conjunction. And one of the other interesting things, this goes back to a little bit what Paige was talking about, is some of the data showing the expression of these different receptors in different portions of the body. So there's some data looking at Tempras2 expression in different brain cell lines, human brain cell lines in vitro, as well as in vivo studies looking at Tempras2 expression in different portions of the mouse brain, and showing that Tempras2 is expressed at higher levels in brain cell lines and brain tissue than ACE2. So this does potentially suggest a role for Tempras2 in some of those neuropathology effects that we're seeing with COVID-19. And again, there are some clinical trials that are currently looking at the effect of a Tempras2 inhibitor, camostat mesylate. So we do have a knockout model of Tempras2 that could potentially be used in combination with one of the ACE2 models that have been previously discussed, and you could potentially look at modulating the progression of disease by using a knockout of Tempras2 with one of these ACE2 models. And lastly, if I've got one more minute, one of the reasons I was really interested in to present this particular model is actually my background in studying Down syndrome. Tempras2 is one of the genes that's found on human chromosome 21, which is trisomic in people with individuals with Down syndrome. And so people with Down syndrome have 3 copies of Tempras2, and this potentially could overexpress the protease, which could have an effect on the infection rate and the progression of disease in individuals affected with SARS-CoV-2 in that population. And I was doing some research to see, and it does seem like there's a lot of reports showing that individuals with Down syndrome do have a more severe progression of disease following COVID-19 infection. So that was a personal connection for me.
Awesome, thanks, Sarah. And just to let everyone know, the stock number for this model is 26196. And I will go ahead and wrap up with 2 of my sort of favorite models that have really not been fully studied at this point in terms of COVID-19. And it's our 2 dipeptidase 4 models, or DPP4. And these models have been used to study MERS-CoV infection previously. So the first model that I want to talk about is the SFTPC DPP4 model. And this has expression driven in the lung epithelia by the human surfactant-associated protein C promoter. And so we see expression in the bronchiolar and alveolar epithelia. And this model leads to a mild infection of MERS-CoV. So it's a non-lethal infection. Now, when DPP4 is expressed in the epithelia that's driven by the human keratin 18 or the K18 promoter that we've been discussing in conjunction with the human ACE2 receptor, expression is observed in the airway epithelial cells, typically where the infection begins. And so this is a model of lethal infection with MERS-CoV. So the really interesting thing I think about these models here is that clinical data has shown us that individuals with diabetes are at a much higher risk for mortality associated with SARS-CoV-2 infection. We know that DPP4 contributes to pathogenesis in type 2 diabetes individuals via cleavage of GLP-1. Studies also show that DPP4 is necessary for MERS-CoV infection and likely as well SARS-CoV-2 infection. So therefore, it's hypothesized that individuals that might be taking DPP4 inhibitors for the treatment of type 2 diabetes might actually have better clinical outcomes for COVID-19 infection than those that do not take DPP4 inhibitors. And so an example of how you might consider using one of these DPP4 models, is to take the DPP4 model and cross it with one of our HACE2 models so that you could infect the mice with SARS-CoV-2 and then look at the treatment of DPP4 inhibitors. And so the stock numbers for those two strains, for the SFTPC DPP4 strain, that stock number is 33727. And then the K18 DPP4 model stock number is 33726. So let's take a look here and see what questions we have. For the CNS, so the SIN1 HACE2 model, Paige, did you see any information on if they die post-infection?
Yeah, so again, the actual SIN1 HACE2 model has not been challenged, at least in publications, with SARS-CoV-2. In the K18 HACE2 model, they do die post-infection. And they actually found that the neuroinvasive potential of this, that the virus is able to replicate very efficiently in the brain. And so there's really a lot more work to be done regarding viral, invasiveness within the CNS and in the brain.
All right, thanks. So we do have a couple of links listed below the video for you to check out. One takes you to our COVID-19 mouse model platform, where you can look at all of the different models that we have available. We also listed a strain search as our second link that also will link all of our models that are not directly COVID related, that don't necessarily have the HACE2 receptor, but are related for COVID research. So I think that's really all the time that we have for this week. Our next talk is called Let's Talk Cytokine Release Syndrome Research Basics. That will be 2 weeks from today on March the 16th. And we look forward to seeing you on LinkedIn and YouTube. So this is Caitlin saying stay healthy, stay safe, and stay excited about research. Bye, everyone.
Bye, guys.
Bye.
Bye.
Yeah.